All right, so I may have lied in that last video. I only did that glycolysis picture, right? So I had this slightly more complicated version from now in section 5 from section 4. But now I'm going to draw the Krebs cycle in this video. So Krebs cycle starts out with your pyruvates that were produced in glycolysis. And our first step of the Krebs cycle is I'm going to take those pyruvates that have 3 carbons in them and turn them into 2 carbon molecules, right? So if you need to up here, make sure you know that this has 3 carbons in it. But otherwise, 3 to 2, if I'm going to lose a carbon at any point in this picture, I'm always going to lose it as a carbon dioxide. This is where that stuff that you're breathing out right now comes from. This reaction, these reactions in this cycle. So I'm going to lose a carbon right away. My 3 carbon molecule turns into 2 carbon molecule. And I'm going to catch some energy in here because this is catabolic. So I'm going to create some NADH right away. So you remember this NAD plus and NADH from that last picture that we did. It's just a new energy molecule similar to that one from photosynthesis, that NADPH, but it's not exactly the same thing. So you just kind of have to know that there's 2 different ones, right? Don't try and mix and match. But otherwise, my next step is I have to take these 2 carbon molecules and I'm going to combine them with an enzyme called coenzyme A, and that should make a acetyl-CoA, which is really just a complex including these 2 carbons, right? So acid as a prefix means 2 carbons. That CoA grabs onto those 2 carbons and it takes those 2 carbons, and with an existing 4 carbon molecule, you can take 2 carbons and 4 carbons, combine them to make 6 carbons, right? So this is kind of a little goofy picture right here, but I take this coenzyme A, grabs 2 carbons, so I can add it to 4 to make 6. Then that coenzyme is released. I can grab onto another 2 carbons, take those 2 carbons, add them to another 4 carbon molecule, make another 6 carbon molecule. So this is a little mini cycle right here. Coenzyme A is essentially our equivalent to Rubisco from photosynthesis. It's the one enzyme that we're going to talk about because it's the one thing that keeps this thing going, right? That keeps this whole process going. So it's really important for that reason. I'm going to make this 6 carbon molecule right here. That 6 carbon molecule is called citric acid, which is why sometimes this reaction is called the citric acid cycle. But otherwise, my next step in the process, I'm going to take this 6 carbon molecule and turn it into a 5 carbon molecule. That is again catabolic. That also means that I'm going to lose another carbon. So I'm going to make a CO2. But that catabolic reaction is going to power the production of another NADH from NAD+. And then we move on to our next step. 5 carbons is going to turn into 4 carbons. Again, I lost a carbon, so I'm going to have to kick out a CO2. This reaction actually makes more than one molecule, energy molecule that is. So I'm going to make an NADH here from an NAD+. But I'm also going to get to make an ATP molecule here, which we talked about before, right? Krebs cycle actually makes usable ATP. The problem is it only makes 1 ATP per pyruvate. There are 2 pyruvates, there are 2 pyruvates per glucose, right? And when we talked about this a long time ago, right, that means I get to make 2 ATP in Krebs cycle, but I don't really want to make this 2 ATP when I already had 2 ATP to begin with. So this is the same context. We still have the same stuff from before coming into play. This might seem really good that I made some ATP here, but unless I have oxygen available, unless I have oxygen to take advantage of all these other energy molecules that I'm making in this picture, all the NADH molecules, then that ATP that's made is actually pretty insignificant, right? It's not really necessary to make this ATP if you can't use all those other energy molecules. And I'm not even done yet, because this 4-carbon molecule has to turn into another 4-carbon molecule. We're going to put a little apostrophe next to it. In your math, when you put a symbol like that, sometimes you call it a prime. So I have to put a mark next to this 4-carbon molecule to make sure you know it's different than this 4-carbon molecule, but otherwise we don't really have to know exactly what's happening there. But this, again, makes some more high energy molecules, some more NADH, that would be worthless without oxygen. So I got to make this NADH. There's actually one more molecule that's made in here, a new different one. That's called FADH2. So that's going to be made from FAD plus 2. So FAD plus 2 and 2 hydrogen ions can make this FADH2. And all that means is that there are actually 4 electrons that are captured in that higher energy molecule, as compared to the 2 electrons that are captured in NADH. So it's going to be useful. It's even more useful than NADH molecules. But again, I made all these high energy molecules, and unless I have oxygen, I got nothing to do with those, right? So like we talked about before, this Krebs cycle is really, really important. There are a lot of energy molecules being made that could be used to make many, many more ATP in electron transport. But if I don't have oxygen available, I really shouldn't be doing this. Right? So this is actually a complete picture for Krebs cycle. So we can be done with this video for now.